Assalamualaikum warahmatullahi wabarakatuh. We are from Group 9 and we are going to present the topic Metabolism of Intestinal Cysteine. The group members consist of Nur Shakira binti Abdullah, Nur Izalia Shadia binti Ashimi and Nur Inshira binti Muhammad Rasiki. Introduction. Cysteine is an amino acid that contains sulfur that is created when two cysteine molecules are oxidized and joined together by a disulfide bond. Cysteine is a nutritionally glucogenic non-essential amino acid. It is created from two amino acids, methionine, which is necessary for nourishment, and serine, which is not. Serine provides the carbon backbone, while methionine provides the sulfur atom. About 20% of the dietary methionine intake, which is mostly transmethylated to homocysteine and transsulfurated to cysteine, is metabolized in the gastrointestinal tract, which is a metabolically significant location of sulfur amino acid metabolism in the body. About 25% of the body's total transmethylation and transsulfuration occurs in the GIT. Additionally, 25% of the diet cysteine intake is used by the gut, which also absorbs 65% of the splenic first pass amount of cysteine. Next, the rules for metabolism of intestinal cysteine. Cysteine provides an essential functional and metabolic significant impact on human health and disease. Cysteine is an amino acid which is a component of proteins that are used by every cell in the body. It is often taken as a supplement in the form of N-acetyl-L-cysteine or NAC in short. This is converted by the body into glutathione, a potent antioxidant, and subsequently cysteine. Now, moving to the synthesis of cysteine. Cysteine synthesis endogenously mainly in the liver, into the lesser extent of the gastrointestinal tract. Cysteine is synthesized from two amino acids, methionine, which is nutritionally essential, and serine, which is not. Methionine furnishes sulfur atoms and serine furnishes the carbon skeleton. Homocysteine metabolized by the gastrointestinal tract from dietary methionine through transmutation condensed with serine that is catalyzed by cystathionine synthase to form cystathionine. Cystathionine layers place and break down cystathionine into homoserine and cysteine and further deaminate by homoserine deaminase to form alpha-ketobutyrate. Sulfur atoms in the polypeptide chain of cysteine could form a bond together with another cysteine, namely as disulfide bridge by oxidation, then forming cysteine. Now, let's move on to catabolism of cysteine. Cysteine catabolism occurs mainly in the liver. It can be catabolized by two different routes, which is transamination pathway and direct oxidative pathway. In the direct oxidative pathway, cysteine is oxidized to cysteine sulfonate by cysteine dioxygenase. Cysteine sulfonate undergoes transamination reaction and gets converted to sulfate and pyruvate under the sulfuration reaction, and then sulfate gets converted to next active form, which is phosphoadenosyl phosphosulfate. Back to cysteine sulfonate. Cysteine sulfonate undergoes decarboxylation reaction and there is a removal of carbon dioxide as byproduct, which is then converted to hypotaurine. Hypotaurine then is oxidized to taurine, which is an inhibitory neurotransmitter that conjugates with bile acid acid to form taurocholic acid. Now, let's look at the transamination pathway. Cysteine undergoes transamination with the act of enzyme cysteine transaminase in the presence of alpha-ketoglutarate to form beta-mercaptopyruvate. And then there is also synthesis of glutamate. Beta-mercaptopyruvate undergoes trisulfuration reaction with the aid of sulfur-transferase enzyme to form pyruvate and hydrogen sulfide. Hydrogen sulfide, with the help of reduced glutathione enzyme, which is tripeptide, is converted into sulfide. The sulfide form in the reaction is oxidized to sulfite and then is oxidized to sulfate. The sulfate thus formed is either excreted in the urine or converted to active sulfate, which is phosphoadenosyl phosphosulfate by using ATP.